receive samples in many different ways. We either receive them dry like this. There's a clip from a fin, so we don't actually have to kill the fish. We can just take a little sample of their fin, and then I will take a little bit of that. So right there is all the tissue I need to get DNA. This is a little bit of soap, a little bit of salt, and this will actually help dissolve all the tissue and release the DNA from each cell. From here I would incubate this for several hours, and then that'll release all the DNA. And DNA makes up everything that we are. Our hair color, our eye color, skin, everything. It codes for all our organs, everything. And this is how we do all of our work. And so from here, after I get the DNA, then I would run what's called a polymerase chain reaction. So the DNA is like this huge long chain of base pairs. And so what we want is, we usually just want to look at one little tiny spot of that DNA. And so what we have here is we have a specific room for setting up our PCR reactions, and that gets us away from all the DNA that we have in the lab, and so we can set up our mixture that we add to the PCR reaction in here, and then bring it out to our DNA. So this is just kind of a clean room that no DNA really comes in here, so we don't contaminate other people's research. We have refrigerators that hold all our reagents, freezers, and then pipettes to make our mixes for our PCR reactions. From there, we take our DNA in our trays, and we come into our equipment room, which we have centrifuges, incubators. We kind of made our own core lab here. We can go all the way through genotyping or sequencing here, small scale sequencing. These machines here are what are called thermocyclers, or PCR machines. And what they do is they are able to go to different temperatures. So to do a PCR reaction, you have to, so you have a double helix, you have to actually separate that double helix with heat. So you take it up to 96 degrees, that separates it out. Then you have your specific primers that will then attach to the DNA at another temperature, and then an elongation step which then copies the DNA in both directions. And so then you go from one copy of your thing, and in over 25 to 30 cycles, you get billions of copies of your DNA. So then at that stage, then you can run it through a machine like this. So this is our DNA analyzer, and we can get data off of that from each size. Yeah. So what we're looking at here is I have the capability of doing 96 well plates, and it goes through this capillary system. So there's a polymer inside each one of these little capillaries that forms little canals, and these canals make it so the DNA has to like wiggle through. The smaller fragments of DNA go through faster than the larger fragments, and just because how they have to wiggle through this maze of polymer. Each one of these rows is a different sample, and within each row you have different DNA fragments. So each different color is for a different fragment that we've amplified. And so these are the same type of machines that they used for the Human Genome Project. and it took them 10 years to do that. Because with this I can only get about 96,000 base pairs maximum with each run. The newer technologies that we utilize on campus, they can actually do next generation sequencing, and where I only can get 96,000 base pairs on this machine with each run, they can get 1 billion base pairs in one run. And so technology has gone much further than this machine allows us to do, but for our purposes, this does what we need.